Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. I hope you're all having a beautiful and blessed day today, inshallah. And in today's video, we're going to talk about the channels of cell membrane. And we're going to talk about two channels of cell membrane. The first one is about aquaporin water channel, and the second one is about ligand-gated ion channels. So without further ado, let's jump into the video.
Hello, good morning. My name is Bara Sharif, and today I will explain the lesson what are aquaporins and what exactly they're doing and what's inside. What are aquaporins? Aquaporins are transparent proteins that regulate the flow of water into the out of cells. Also, aquaporins, often called aquaporins water channels, are a family of small, integral membranous proteins that are expressed broadly throughout the animals and plant kingdoms. They have a similar basic structures, with the aquaporins membranes consisting of 6 transverse helical segments and 2 short helical segments. They surround cytoplasms and extracellular vesicles connected by a narrow aquaporin spore. They contain several conservative, including NBA, acquaintances in their short helical segment, aquaporins, monomers, assemblers, transversions, and membranes, with each member functions independently. Aquaporins, they allow the passage of water. They prevent the passage of ions. If aquaporins allowed ions throughout their channels, all ions concentration gradients across the cell's membranes would approach zero, and the cell would not be able to perform many of critical functions that depend on contractions gradients of virgin muscles. To understand how aquaporins regulate water movement into a single file and prevent the movement of other ions, it's important to understand the three-dimensional structure of the aquaporins proteins. An aquaporins channel is composed of 6 transverse helicals with both of amino and carboxyl transverse. The cytoplasm side that membranes 2 of the 5 loops that contact by 6 transverse helicals, and they're extremely hydrophobic. One of these loops is the instructional side of membranes, and the other is one of extractional side of membranes. For aquaporins, channels associated with each other is the membranes, so that the one location there are 4 possible passageway of water into move throughout the cell's membranes. Each aquaporins channels can have a slightly different proteins structure. There are at least 4 different aquaporins in mammals, and I think about 14 approach channels found within plants. It's important to remember that aquaporins don't actively transport water across the cell's membranes. Instead, they facilitate the diffusion of water across the cell's membranes. Due to the slow diffusion of water across the lipid layers, aquaporins effectively increase the overall rate of water movement across the cell's membranes. Thank you.
And next, we're going to talk about the diseases and disorders of aquaporin channels. So the first one is neuromyelitis optica, or NMO, or also known as Davis disease, is an immunological disorder characterized by inflammation and demyelination of the eye nerve and the spinal cord. The most specific marker of the disease is the presence of IgG antibodies against aquaporin 4. This disease is a very rare condition and is often misdiagnosed as multiple sclerosis or thought to be a type of multiple sclerosis. Since it is an autoimmune disorder, meaning that the body's immune system reacts against its own cells in the central nervous system, specifically, in this case, in the optic nerve and the spinal cord. And the existence of antibody called neuromyelitis optica immunoglobulin G, or NMOIgG, in the blood may damage aquaporin 4, which is the water channel surrounding the optic nerve and the spinal cord cells, causing the inflammatory effect of the condition. The condition may sometimes appear after an infection or may be associated with another autoimmune condition. And the people with NMO usually experience symptoms such as weakness or paralysis in arms or legs, blindness in one or both eyes, numbness or loss of sensation, painful spasms, uncontrollable vomiting and hiccups, bowel or bladder dysfunction from spinal cord damage, and children might experience confusions, seizures, or coma. And the next one is nephrogenic diabetes insipidus, or NDI, is an inability to concentrate urine due to impaired renal tubule response to vasopressin, or ADH, the antidiuretic hormone, which leads to excretion of large amounts of dilute urine. So simply, the kidneys produce a large volume of dilute urine because the kidney tubules fail to respond to ADH, and the kidneys are unable to reabsorb filtered water back into the body. NDI can be either acquired or hereditary. About 90% of all cases of hereditary NDI result from mutations in the AVPR2 gene. Most of the remaining 10% cases are caused by mutations in the AQP2 gene. At least 40 mutations in the AQP2 gene have been identified in people with NDI. Both of these genes provide instructions for making proteins that help determine how much water is excreted in urine. So, AQP2 gene mutations cause the aquaporin 2 protein to be misfolded into an incorrect three-dimensional shape. The misfolded protein is trapped within the cell, where it is unable to reach the cell membrane to transport water molecules. Well, a few mutations result in the production of functional aquaporin 2 water channels, but these channels are misrouted within the cell and do not reach the cell membrane. And then if aquaporin 2 water channels are not inserted into the membrane of collecting duct cells, the kidneys are unable responding to signals from ADH. As a result, the collecting ducts don't reabsorb water as they should, and the body makes excessive amounts of urine. And this is the picture of NDI. Maybe you could explain a little bit.
Assalamu alaikum warahmatullahi wabarakatuh. Hi everyone, I hope you are doing well. So, have you ever wondering why our brain, our system nervous reacts so quickly? So, this is our topic today. So, there is a ligand-gated ion channels, and these are kind of receptors that react to binding of a ligand. A ligand is a chemical messenger. So the procedure is, or as you know, this kind of receptors are there in our cell membrane. So they facilitate the transport of ions. As we know, the procedure is that there is a ligand comes into the channel, then it happens a combination or a combination. Yes, that's right. Then it's like a key and a lock. The receptor opens, then the ions come into the cell or out of the cell. So this is the process, and these kind of receptors are commonly found in electrically reacted cells because it's important to react so quickly. So this is the explanation. If you have any question, please ask. Thank you.
Assalamu alaikum wa rahmatullahi wa barakatuh. So I will speak about cases and abnormal conditions happen to ligand-gated ion channels. So I will speak about two receptors. First, nicotinic receptors. Actually, they are ligand-gated ion channels which modulate cell membrane potentials. And the mutations happen to it. They are associated in two syndromes. First, the fast channel syndrome, and second, the slow channel syndrome.
And here I'm going to add a little bit about the explanation. So as mentioned before, fast channel syndrome and slow channel syndrome are associated with mutation in the nicotinic receptor, while nicotinic receptors located at the neuromuscular junctions of somatic muscles, and they are responsible for muscular contraction as well as the neuromuscular signaling. And patients with this mutation usually present at birth with muscle weakness, rapid fatigue, and progressive muscle atrophy. Here I'm going to add a little bit about the explanation of what is actually happening in slow channel syndrome and fast channel syndrome. So in slow channel syndrome, mutant AChR channels have increased activity and longer channel openings, producing a prolonged depolarization of the muscle cell membrane, and this leads to excessive depolarization results in inactivation of sodium channel, accounting for muscle weakness. And in contrast to slow channel disorder, the channel either failed to open or close too quickly in fast channel syndrome. Mutations in FCS appear to decrease binding affinity of acetylcholine and the ability of the channel to open.
Secondly, I will speak about glycine receptors. Actually, they are members of CYS low family of ligand-gated ion channels, and also they are responsible for mitigating the inhibitory effects of glycine. Mutations in this channel are associated in a very rare disorder called startle disease.
So I'm going to talk a little bit about the startle disease, only some additions. And as stated earlier, startle disease is associated with glycine receptor mutation, and glycine receptors are widely distributed throughout the central nervous system, or the CNS, particularly within the hippocampus, spinal cord, and the brainstem. And startle disease is an autosomal dominant disease that causes stiffening of the muscles in response to startling. Severe responses can occur along with injuries, as the rigidity prevents patients from reaching out when falling. So I think that's all from us about the two channel of the cell membranes, and thank you so much you guys for watching the video. I hope this will bring benefits. Assalamualaikum warahmatullahi wabarakatuh. Have a great day.